Now, in part 3 of the Intro to Medical Genetics, we're going to build off the first two parts, which, one, introduced where the genetic material is housed and how it's housed in the cell. Two, how that genetic material in the cell is propagated and replicated as generations go on. And now we're going to talk about how the cell actually uses DNA at the molecular level. And this really brings us to the central dogma of molecular biology, where, as we can see, starting with DNA, that is transcribed to RNA, and that is translated into protein, which goes on into the body to basically exert the gene's action. Now, there are some exceptions to the central dogma, which we won't talk about in this class, but for our purposes, the central dogma is really kind of the end-all, be-all. It's where we're going to concentrate our time. So first, we're going to look at replication of DNA in the cell. So what is DNA replication? It's really that replication or that increase of genetic content in the S phase that we talked about previously in the propagation of DNA, preparing for mitosis and meiosis. It's copying the DNA in the cell so that it can be, again, divided and propagated throughout the body or into gamete productions and things like that. So how does this work? Essentially, this occurs at any point in the DNA. You can have replication occur at different DNA, at different genes throughout your entire DNA. That's what's called DNA replication forks. And why are they called DNA replication forks? And that's because the actual DNA itself ends up looking like a fork. So let's say we have some DNA. And down here it's double-stranded. Okay? And then it's split apart here. Okay? So, all DNA has what's called a 5' and a 3' end. And that's really just suggesting you, or at least telling you, what direction the DNA flows in. And DNA always flows in downhill, as we say. It always flows from 5' to 3'. That's always the flow of DNA, kind of its natural direction. So a 5' to 3' DNA is called sense, and a 3' to 5' DNA is called antisense. And sometimes you might see this in certain pharmacogenetic literature, so it's good to have an idea of what it is. So for our DNA here, our 5' end would be, this would be our 5' end and the associated 3' end, while this would be our 5' end and associated 3' end. And so when helicase comes in here, and that's a special protein, let me change the color on this. There we go. That's a special protein. We'll make proteins purple. And that's called helicase. This is the actual protein that unwinds DNA and creates the fork, the DNA replication fork that we're talking about. And so it unwinds it, and you end up having a leading strand up here and a lagging strand here. And why do they call it a leading and a lagging strand? It's because that is how DNA can be replicated on each strand, because DNA replication actually ends up producing two identical double-stranded DNA molecules. So what comes down here again is another protein, and this protein is called DNA polymerase. And on the leading strand, because of the way DNA is actually made, it can make a continuous strand of new DNA going from the 5' to the 3'. So just remember, DNA polymerase always likes to head downhill 5' to 3', and the reason it's called a leading strand is because the DNA polymerase can go 5' to 3' off of the DNA's parent strand, which is actually going 3' to 5'. So it has to make the DNA downhill, but it's actually kind of going uphill on the parent strand. So how does DNA polymerase do this? It incorporates those letters we talked about before, adenine, cytosine, guanine, and thymine. It brings those in so that if on the leading strand you had, let's say for example, A here, T here, G here, C here, T there, you would have the matching by DNA polymerase of an A here, a G here, a C here, A here, and a T here. Because we remember, A pairs with T, G pairs with C. And that makes a continuous, nice strand to make a new double-stranded DNA molecule. However, on the lagging strand, we have a bit of an issue. And that's because DNA, to be replicated, has to head in a downstream nature, right? So to head downstream, it has to go this way in order to go 5' to 3'. And so how the cell does that is it does it in these little chunks, and these chunks are actually called Okazaki fragments by the researcher who discovered it. So DNA polymerase comes in just like before, DNA polymerase. However, it starts it in sections, okay? And so for each section, it starts to head 5' to 3', essentially until it runs out of room. It interrupts, or it gets kind of kicked off the parent lagging DNA strand by the other DNA that's already there. And then an additional protein comes in called ligase. What ligase does is it comes into these gaps that are made, and it uses its own proofreading function to actually connect these together, eventually making a single DNA strand that's base paired with the original parent strand. So in the end, this continues because the helicase continues to head this direction, unwinding the DNA. On the leading strand, DNA polymerase just continues to head down. On the lagging strand, more sections are added on, that is ligase together. And the end result here is you end up having 2 copies of your DNA that's double-stranded. One that resulted from the lagging strand, and one that resulted from the leading strand. And there you have DNA replication. Now let's take a look at a video similar to the ones we've seen before that kind of shows a graphical representation of what DNA replication would look like inside a cell in real time.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
Okay, so you can see there it showed both the leading strand replication and the lagging strand replication to create two identical copies of DNA. So let's do a knowledge check here. If this is a base sequence of a strand ACT, CGT, what would be the base sequence of the complementary DNA strand? So let's work this out. ACT, CGT. So it would be, if this is the parent quote-unquote strand, what would be the base sequence of the complementary strand? And basically this is, we already did this. We have to remember that an adenine always binds to a thymine, a cytosine always binds to a guanine, and then we fill it out from there. The answer is TGA, GCA, which is letter B. As you can tell, letter E, answer E, is not the correct answer, don't know, and answer C and D actually have uracils in them, which are only found in RNA, which we're going to talk about next. So coming back to the central dogma, now we've talked about replication, and we're going to shift our focus on how DNA is actually expressed, the beginning of DNA expression, which is the transcription into RNA. We do need to clarify that there are several types of RNA that are used in the cell. We'll talk about three main types today. However, I will put the caveat that there are many, many more interesting types that have been discovered recently that have primarily epigenetic purposes. However, we won't talk about them today. So today we're going to talk about, though, is mRNA, which is primarily found in transcription, and then we'll briefly mention rRNA, which is a RNA that makes up ribosomes used in translation, and then tRNAs, which actually carry amino acids to the ribosome machineries to actually help translation, and you can actually see that over here as well. But for right now, we're going to talk about mRNA, or messenger RNA. So how does the body know where to transcribe DNA? And it occurs in three major stages: binding, elongation, and termination. So stage one is the binding and initiation of DNA. So essentially how this happens is you have a double-stranded DNA, and so I'm going to make my double-stranded look like this for the purposes for the ease and the purposes of being able to see it instead of making it wounded DNA. You have a 5' and a 3' on your leading strand. You have a 3' and a 5' on your lagging strand. And what's important is that the machinery for transcription actually recognizes a very specific area of DNA, and I'm going to highlight it green. And this specific area of DNA is actually called a promoter because it promotes transcription. So what ends up happening is you have a protein called RNA polymerase, different from the DNA polymerase, but essentially does the same exact function. This recognizes a promoter and binds to it. So what we'll have is an RNA polymerase bind to the promoter region. And then what happens, if we redraw this one more time, is that you have slightly bulged out DNA by various enzymes, including RNA polymerase, mainly does this itself. It begins to unwind the DNA, so RNA polymerase is still docked onto this region. It begins to unwind the DNA, and then it begins to create one strand of RNA heading downhill. So what it's going to do, it's going to begin to make RNA in this direction, because again, it always wants to head downhill, 5' to 3'. If we remember, this is the 3' to 5' strand, okay? And I may have misspoke. This is actually would be technically the leading strand here, right? Because it's able to lead the RNA transcription downhill in one continuous strand. So a difference here with RNA is that there are not two replications taking place, or transcriptions, I should say, taking place. There is no lagging strand RNA made, because RNA is only a single-stranded molecule, so it only needs to occur on one strand. And so this further continues to make a transcript. But this is where the binding and the initiation take place. Sometimes the promoter region will actually have specific letters in them. A common one that's taught in the literature is the ta-ta box. So there's specific letters, including a long repeats of TATA, and really it's just a recognizing sequence for RNA polymerase, which goes down and binds. And there's other factors called transcription start sites factors, or transcription factors. And there are other proteins as well that bind on and essentially just stabilize the RNA polymerase onto the DNA so that it can begin to pull in the different letters, which again, if we remember, is A, C, U, because uracil replaces the thymine, and G in RNA. This brings us to the second step of DNA transcription, which is just basically elongation. So if we go back to our picture of the previous slide where we had a double-stranded DNA that has been docked by an RNA polymerase here. It's a big molecule that kind of grabs onto it. What's occurring, and what we kind of visualized in the last slide, is RNA polymerase is now just elongating the RNA downstream by grabbing all those relevant RNA-based nucleotides from the cytoplasm and adding them in here. So elongation essentially is just matching based off the DNA. So we can do an example of that in here. If we have an A, T, G, A, C here, it's going to bring in letters to match those, right? Except for the only catch here is that the thymine is changed to uracil. So instead of a thymine here, you'd have a uracil, then you'd have an A, adenine, or an A, you'd have the cytosine, and back again you'd have a uracil, and you'd have the guanine. And those actually, generally they might make some temporary bonds here, but eventually this is going to peel off because the uracils have an unfavorable bond, and that promotes the peeling off, which we're going to talk about the next step, which is termination of RNA transcription. So how does the termination of RNA transcription or DNA transcription into RNA actually occur? Again, we have our double-stranded DNA here. And this time what I'm going to show you is essentially two different versions that tells the RNA polymerase that you've done your job, it's time to get off of the DNA. Because the RNA polymerase needs to know this, because generally RNA polymerases are only transcribing certain genes or sections of genes at a time. They're not doing more than that. So in here we would have, for example, an RNA that this RNA polymerase has made, 5' to 3', and when it gets to the end, it needs to get it needs to know that it's time to get off by two different methods. And those two different methods are either a CG-rich region or a Rho protein. So what happens in a CG-rich region? Essentially, what's going to happen is that at the end of transcription, it becomes very CG-rich in this section here of the RNA. And this actually causes the RNA to loop itself on the DNA molecule. So it starts to pinch off. And the reason that happens is because the actual attraction for its own nucleotides are so strong that it begins to pinch itself off here. And eventually what will happen is the pinching will become so strong that it'll pull itself off, and you'll have your single-stranded floating RNA, mRNA transcript, and then your double-stranded DNA still exists in solution as well, or in the cytoplasm. So that is the CG-rich region. Again, basically just saying that the end of an RNA transcript is so CG-rich that it causes a loop to form in the RNA itself, and then it becomes unfavorable for the RNA polymerase to stay on there. It kicks off, and then the rest of the RNA molecule pulls off the DNA as well, because the DNA wants to go back to being double-stranded with itself. That's the most energetically favorable kind of setting that it wants. The other way is something that's called a Rho protein. And a Rho protein is essentially just a termination protein that comes along a DNA or an RNA transcript, 3', 5', and it's a Rho protein, and it kind of scans along the mRNA transcript until it gets to an area that it recognizes as a termination area. And then what happens is it actually pushes off the RNA from the DNA itself. So it scans along until it finds its recognition sequence, and then it actually is able to call in other proteins and kick the RNA polymerase off, and you get the end result, which is the same as the other side. You have your RNA, mRNA transcript that's now available to be translated, and then the DNA goes back to being double-stranded again. So these are the two main mechanisms of DNA transcription termination. Now let's watch again this visualization of the transcription, initiation, elongation, and termination.
Central dogma of molecular biology: DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
So you can see there, again, what's really interesting about these videos is the rapidity which things are made within the cell and how quickly it happens. But you could see the initiation, elongation, and eventual termination of the RNA transcript based off that one side of the DNA. So let's do another knowledge check. In what direction is the RNA polymerized or made? Is it A, in the 5' to 3' direction, B, in the 3' to 5' direction, C, downstream to upstream, or D, depends on what DNA strand the RNA polymerase binds. And, as we know, and I continually said, for both replications of DNA or transcription of RNA, the body really favors that upstream to downstream replication or transcription of RNA. So this is how RNA is transcribed. It's always downhill to end up making a 5' to 3' mRNA transcript. And with DNA, it's able to do it on both strands. However, it's much easier for it to do it on the strand, the leading strand, that makes the DNA in the 5' to 3' direction. So always downhill when the DNA or RNA polymerase makes the new strand. That's very energetically easy and favorable for it to do it. And so for an RNA that only needs one single strand, it's always 5' to 3'. So, in the central dogma, we've gone over the replication of DNA, the transcription of DNA into RNA, starting the process of gene expression. Now we're to the final step of translation of RNA into protein, which then have functions that we think about and we talk about in our pharmacology classes all the time. The enzymes that chew up drugs, the transporters that transport in drugs, those are proteins. However, before we get to translation, we need to talk about pre-translational modifications. And these are pretty specific and explicit in RNA. So let's take a look and see what happens. So we started with DNA, and I'm just going to do one line, but you can assume this is double-stranded DNA. And we have DNA that looks like this. Has a 3' end and a 5' end. And these solid and dotted lines are referring to parts of DNA that are called exons and introns. And exons and introns become important in the pre-processing of RNA into its final form. So we talked about transcription. And what we ended up with, even though this is reversed, we ended up with a single-stranded RNA that perfectly copied, except for changing the T's to the U's, if we remember that. And so we have here, we still have the capturing of exons and introns. And what we have is an unprocessed mRNA, unprocessed RNA transcript. So what do we need to do to process it? First, we have to add in what's called a methylguanine 5-degree cap. And so what that looks like is just like this. We have 5' methylguanine is added to the 5' end. Oop. Let me get rid of that. There we go. Is added to the 5' end. But we still have all of our exons and introns. And so now we have a partially processed mRNA, which has a 5-degree cap added on the end. So this step adds a cap. But we need to process it further. And the next step here is where the introns come out. So now the introns are taken out and the exons are spliced in. And the way I always remember it, it's the opposite of what you would think. The introns are out, the exons are in. So what we end up with is 5-degree methylguanine cap, and then a nice solid piece of processed RNA that only contains exonic RNA, which is based on the exons of DNA. And another way we could say this is an mRNA that only contains the coding regions of DNA. Finally, to do the last preparation step, we need to add in a poly A tail. And so what that looks like is we have our 5' with our methylguanine, and that with our exonic RNA, and then at the 3-degree end we add a very long poly A tail. And what this does, specifically the 5-degree cap and the poly A tail, is it stabilizes the RNA molecule and allows it to be transported out of the nucleus and into the cytoplasm, because in the cytoplasm is your translation machineries, aka your ribosomes. And that is how we prep an RNA to be transported out of the nucleus and get it ready for translation. So let's take a look at that again with our little graphic representations.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
So I think that video does a real nice job showing you how it's processed, and specifically, and what I forgot to mention here, is that with the splicing events, these are facilitated by that enzyme that they were talking about there, the spliceosome. So that actually brings it together. As you can see, it brings together those two recognition areas, because the beginning and the end of introns and exons, they have specific recognition areas that the protein that you saw kind of loops it together, and it creates an event that actually is able to pop off the introns in that loop that we saw and put together the exons in kind of a single shot. So that's how that works. So let's do another knowledge check. If you were to investigate the effect of a genetic variant on drug response of the following, which would most likely affect the protein's function? So would it be a variant located in a region upstream to the gene? Would it be a variant located in the exonic portion of a gene? Would it be a variant located in the intronic portion of a gene? Or maybe a variant located in an adjacent gene? So again, we're to investigate a genetic variance effect of a genetic variant on drug response. We want to know which variant, where in the gene it would most likely have an effect on the protein's function. And so the best answer here is B, because we just learned that the introns go out and the exons stay in. And remember, another name for exonic DNA is coding DNA. So a lot of times in papers that you guys may read or that you'll present on at the end of the class, you'll read about, well, this SNP was an exonic SNP, this SNP was an intronic SNP. And so in general, researchers are much more excited, or at least have more theoretical basis, if they find a SNP in an exonic portion. However, that's not to say that intronic SNPs aren't important, and we'll talk about some of those reasons later in the class. So now we've reached the final steps here. So we've gone over the replication of DNA, the transcription into an RNA, which is now pre-processed, and now we have a pre-processed RNA, or mRNA, that is ready to be made into an actual protein. So one of the unique parts of translation is how does the machinery within the cell actually read an RNA to create a polypeptide and eventually a protein. And the way it does this is it reads it through what are called codons, or letters in 3 off of RNA. So the machinery will come, the ribosome, and it'll read every 3 letters with a start codon, and it'll match those to a tRNA carrying a methionine. That's a start codon, and it'll read those every 3. And you can actually have what's considered a codon chart, where you know that if you have a UUU or a CUU on an RNA or an ACU, these code for certain amino acids that are added on to the polypeptide to eventually make a protein. Now, codon charts can be different types of codon charts. You can have an RNA codon chart. You can have a tRNA codon chart. They're all effectively doing the same thing, which is reading different groups of 3 letters to add on a amino acid, but you really need to know what type of chart you're looking at in order to translate effectively DNA into RNA into amino acids, which make a protein. So that's how the machinery does it. It's very clever, and it's redundant in and of itself. And the redundancy is nice because when you need to make RNAs and proteins very quickly in the cell, you don't have time to proofread them to a significant extent. So it kind of allows for some play in the letters, but still getting the right amino acids on the back end. So let's take a look at what translation actually looks like. So we have our processed and ready to go RNA. Remember, it has the poly A tail, it has a 5-degree cap here, and it's basically ready to be read by a ribosome. So a ribosome kind of looks like a big protein. It sort of looks like this, and what it does is it sandwiches in the RNA and tickertates it, if you will, to read it. And so what it does is it shoots along the RNA in this direction and reads it. So for example, we could have an RNA that has an AUG, a UUU, and maybe a CGA. And what it does, it'll start here, it'll go along until it finds a so-called start codon. So our ribosome started on the RNA, and it finally got to its first start codon here, and that makes it call in tRNAs that are floating around. And remember, we talked about tRNAs, and those are basically RNA molecules. And the reason they're RNA molecules is because they have an anticodon that reads onto it. So you would have to have here UAC. What happens is that the ribosome tries a bunch of different tRNAs that are floating around the cytoplasm, and then it finds one that actually works with the AUG, and attached to the AUG is an actual amino acid, and in this case it's methionine. So what happens is that it begins to add these on one by one. If we continue to draw this in, so let's pretend that the ribosome is still there. So on your AUG, you actually have the UAC, that's wrong, the wrong color, UAC, tRNA attached to a methionine. And then the ribosome reads the next three letters, which in this case are U, and it keeps looking for tRNAs that will match the necessary letters, which would be, in our case, what would it be? It would be AAA, and an AAA happens to be attached to a phenylalanine. And this process continues as it goes down and continues to read CGA. So let's do it one more time down here. You have a 5' 3'. So far we've had our ribosome add on for the two codons AUG, UUU. And what happens is that this tRNA leaves. So this one begins to float away, UAC. Okay? This tRNA is still attached. However, what's different is now the phenylalanine is attached to the methionine from before. So these two end up attaching, and that's starting your polypeptide chain. And then this continues down CGA, and that'll look for a GCU tRNA, and that's attached to an arginine. And then that'll continue, and these two will eventually attach together, lengthening your polypeptide chain, which eventually becomes a protein. And what's the difference between a polypeptide chain and a protein? Proteins have very, very complex secondary, tertiary, quaternary structures that actually make them be an enzyme or a transporter or whatever the protein's function may be. The ribosome will keep working its way as it goes down the line until it reads a typical stop codon. I put a couple here, so let's say way down the line you have a stop codon, and at that point the ribosome will call in the last tRNA, and then the ribosome will dissociate from the RNA. The RNA generally dissolves at that point, and all you have left is your polypeptide chain, which goes on to do its protein functions. So let's see that in real time again.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides, or 1 codon. Thirdly, the spent tRNA is moved to the E site, and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
So I think that video again shows you nicely about how the tRNAs are floating around in the cytoplasm, how the ribosome essentially faster than probably even we could process, the human eye could process, is trying different tRNAs for a given codon until it finds the right one, and then it begins to assemble the polypeptide chain into a protein. So true or false? A single codon encodes each amino acid. What do we think? True or false? A single codon encodes each amino acid. The answer here is false. And why is that? Again, that's because of the redundancy that we talked about before, where you can have multiple different codons coding, for example, for a phenylalanine. And remember, that helps the lack of proofreading that occurs to create RNA from DNA and allows certain errors to not really be actually functionally significant. This is something that's important and something that we think about in pharmacogenetics as well. So finally, let's just see a review of the central dogma in this short video.
The DNA double helix contains 2 linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the 2 strands. The yellow chain sneaking out the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the 2 exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it sneaks out into the outer part of the cell. Then all the components of a molecular factory called the ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds, the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Okay, so there's a review of the intro to medical genetics, and really this was just your basic introduction to those features of DNA and how they're propagated from generation to generation, and then actually how they're utilized within the cell for replication and transcription. And really, these are all very important for the future lectures that we'll talk about with pharmacogenetics. Having this background will allow you to better understand and comprehend the different types of genetic variants we're going to talk about.